In the Namib Desert, one of the oldest and driest places on Earth, there are mounds that rise 3 meters out of the sand. They have no architect, no foreman, no blueprint. The creatures that built them are blind. Each one is roughly the size of a grain of rice, and yet the mound they built together are the precision-engineered structure with internal ventilation shafts, fungal gardens, royal chambers, and a climate control system so effective that the temperature inside never wavers more than 1 degree from optimal. No matter what the desert does outside, the builders are termites, and we have misunderstood them for a very long time. We call them white ants. We think of them as pests, as destroyers, as the thing that quietly hollows out the floor beneath your feet until one morning the boards give way. They are the insect we treat with poison, the creature we call an exterminator to eradicate. Mindless, destructive, ancient in the worst sense. A leftover thing from a simpler world. Every piece of that picture is wrong. The termite is not an ant. It is not even closely related to ants. It is not mindless. It does not simply destroy. It is, in the strict ecological sense, one of the most important recyclers on land. A creature whose work makes the soils of the tropics function, whose mounds outlast the civilizations built near them, whose colonies represent one of the most sophisticated examples of collective intelligence in the animal kingdom, and the story of where termites actually came from, how they became what they are when the social colony first appeared, and what we almost got catastrophically wrong about their place in the tree of life. That story has been rewritten completely in the last 15 years by ancient DNA. This is what the genome actually shows. Before we get into it, hit that subscribe button. Every video here digs up the real origin of something you thought you knew. Let's begin. The old story of termite origins was, in the generous reading, incomplete. In the less generous reading, it was a story built on a foundational misidentification that misdirected a century of research. For most of the 20th century, termites were classified in their own order, the Isoptera, meaning equal wings, a reference to their 2 pairs of nearly identical wings in the winged reproductive form. This classification placed them as a separate and ancient lineage of insects, related distantly to cockroaches, somewhat distantly to mantids, and only very loosely to anything else. The assumption was that the social colony, the queen, the workers, the soldiers, the whole elaborate architecture of termite society, had evolved once, deep in the past, in a single ancestral lineage that then diversified into the roughly 3,000 species we know today. The textbooks showed a clean family tree. Termites here, cockroaches there, a respectful distance between them, and a long independent history for each. There was just one problem. When researchers began looking closely at the anatomy of one particular group of cockroaches, a small, dampwood-dwelling genus called Cryptocercus, found in mountain forests of North America and Asia, they kept finding things they did not expect. Cryptocercus cockroaches live in family groups inside rotting wood. They digest that wood using symbiotic gut microbes, specifically a community of flagellate protists that break down cellulose. The parents actively care for their young, feeding them through a process called proctodiotrophallaxis, which means the young eat material passed directly from the parents' hindgut. The family unit is stable across generations, and the colony is defended. Sound familiar? It sounded familiar to the researchers too, but the implications were too disruptive to accept easily, because if Cryptocercus and termites were doing all of these things, these deeply social, cellulose-digesting, hindgut-sharing behaviors from a shared ancestor, then termites were not a separate ancient order at all. They were social cockroaches. The DNA confirmed it, and when it did, it didn't just reshuffle a classification. It changed everything we thought we knew about when, how, and how many times the most complex social behavior in the insect world had evolved. In 2007, a landmark molecular phylogenetic study and the cascade of genomic work that followed it settled the question definitively. Termites do not sit beside cockroaches on the tree of life. They sit inside cockroaches. The order Isoptera was formally dissolved. Termites were reclassified into the cockroach order Blattidia. The family tree was redrawn, and the textbooks, slowly, began to catch up. But the reclassification was only the beginning of what the DNA revealed, because the deeper researchers looked at the termite genome and the genomes of their closest cockroach relatives, the stranger and more layered the story became. The first major revelation was the age, when molecular clock analysis was applied to the termite lineage, anchoring DNA mutation rates against the fossil record. The results pushed the origin of termite social behavior much earlier than the fossils had suggested. The earliest termite fossils, clear unambiguous fossil termites with their distinctive morphology, date to the early Cretaceous, roughly 130 million years ago. For a long time, this was taken as the origin point. Termites appeared in the Cretaceous. That was when the colony began. The molecular clock said otherwise. The genomic data pointed to an origin of the termite lineage, meaning the divergence of the social wood-eating ancestor from the broader cockroach group, somewhere between 170 and 200 million years ago. In the Jurassic, before the great radiation of flowering plants, before most modern bird groups existed, before the dinosaur world had reached its peak diversity, the colony, in other words, began in a world we can barely imagine. In a Jurassic forest of conifers and cycads and ferns, something that looked like a cockroach began living differently. It stayed in the wood. It shared its gut microbes with its offspring. It built a family that didn't leave. And somewhere in the slow accumulation of those behaviors across millions of years, the colony emerged. The gap between 130 million years in the fossil record and 170 to 200 million years in the genome represents something real and important. Those earliest termites were living in a world where their bodies did not preserve easily. Wood is a poor fossil medium. Soft-bodied insects in rotting logs leave almost no trace. The fossils we have are the lucky survivors. The colony existed, the DNA tells us, for tens of millions of years, before we have any stone record of it. The second revelation was the gut. Termite social behavior, inseparable from what lives inside a termite's gut. The flagellate protists that break down cellulose in the hindgut of lower termites are not just digestive aids. They are the reason the colony works. Without them, a termite cannot eat wood, and without the colony, to pass those microbes from parent to young, through that shared hindgut feeding, each new generation would be born unable to digest its own food source. The social colony and the gut microbiome evolved together. The genomic studies showed that the relationship between termites and their hindgut protists is one of the most tightly co-evolved symbioses in the insect world. When researchers sequenced the genomes of the gut protists alongside the termite genomes, they found that the two had been co-diversifying for an extraordinary span of time. The protist lineages inside different termite families match the evolutionary tree of the termites themselves, a phenomenon called cophylogeny, meaning parasite and host have been evolving in lockstep. This means the origin of termite sociality was not purely a behavioral event. It was simultaneously a microbial event. The colony emerged not when one insect became social, but when an entire miniature ecosystem, host and symbiont together, reorganized around a new way of living. The third revelation concerned the soldiers. Every termite colony has soldiers. Depending on the species, they may have massive crushing mandibles, or a long nozzle on their head that fires a sticky toxic secretion at enemies, or a head shaped like a plug that blocks a tunnel entrance with their own body. Soldiers cannot feed themselves. They are entirely dependent on workers to feed them. They exist for one purpose only, to defend. For a long time, the evolutionary origin of the soldier caste was treated as a straightforward story. Soldiers evolved because colonies needed defense, and natural selection favored colonies that had them. Simple. The genomic data complicated this picture significantly. When researchers examined the gene expression profiles of soldiers across different termite lineages, they found something unexpected. The soldier caste, across many different termite families, shows strong molecular signatures of genes associated with juvenile hormone pathways, the same hormonal system that controls metamorphosis and development throughout the insect world. The soldier appears to be, at the molecular level, a permanently arrested juvenile, an insect whose developmental program was interrupted and locked into an intermediate state between larva and adult, repurposed by the colony into a specialist defender. The gene networks that in other insects would push a larva toward reproductive maturity are in the soldier modified to produce weaponry instead. And the most unsettling part is that this arrest is irreversible. A soldier termite cannot become a worker. It cannot become a reproductive. It is locked into its role from the moment the hormonal switch is thrown. Evolution had taken the developmental machinery of an insect and used it as a raw material to sculpt a cast. The fourth revelation is the one nobody saw coming, and it concerns what happens when the queen dies. In most termite species, the colony has a single queen. She can live for decades. In some species, documented queens have survived for 45 years, making the termite queen one of the longest-lived insects on Earth. During that time, she may lay tens of thousands of eggs per day, her abdomen swollen 200s of times its original size. Immobile, tended constantly by workers, when she dies, the colony faces catastrophe. The source of eggs is gone, and in the textbook version of Termite Society, this means the colony will eventually die too, unless a replacement reproductive can be found. But the genomic studies revealed something that overturned this expectation completely. In many termite species, when the queen dies, workers undergo a transformation, not a physical metamorphosis in the traditional sense, but a profound reprogramming. Worker termites that should have been sterile for life begin producing eggs. Their ovaries, which had never been active, activate. Their behavior changes. The colony reorganizes around them. The trigger for this transformation was found in the genome. Researchers identified specific gene expression networks that are suppressed in worker termites by pheromones produced by the living queen, chemical signals that reach every worker in the colony and hold their reproductive potential permanently in check. When those signals stop, the suppression lifts, the gene networks activate, and the colony reasserts itself from the inside. The queen is not just a reproductive, she is a living chemical broadcast tower. Her body is constantly running a molecular program that defines what every other termite in the colony is permitted to become. And the genome of every worker carries the full genetic potential for reproduction, waiting silently beneath the suppression, for decades if necessary, until the day it is needed. Now here is the part of the story that carries the darkest weight. Termites are among the most ecologically important organisms on land. In the tropics, they are responsible for the decomposition of a significant fraction of all dead plant material. Their mounds aerate and enrich soils. Their tunnels allow rainwater to penetrate deep into the earth rather than running off the surface. In some African savannas, the distribution of termite mounds determines where trees grow, which determines where animals graze, which determines the entire structure of the ecosystem. Remove the termites and the landscape reorganizes, and yet termites are disappearing. A global analysis published in recent years found that termite populations and diversity have declined significantly in regions affected by agricultural intensification, deforestation, and soil degradation. The wood-dwelling species, which include the most ancient lineages, the ones whose genomes carry the deepest evolutionary record of how the colony first emerged, are particularly vulnerable. They depend on intact forest with abundant standing dead wood. As forests are cleared and logged, the complex microhabitats those species need disappear with them. The higher termites, the ones that build the great mounds of the tropics, depend on intact savanna and grassland systems. As those systems are converted to monoculture agriculture, the mound builders lose the ecological context that makes their engineering meaningful. We are not just losing termites. We are losing the deep genomic record of how social life in insects actually began. The most ancient termite lineages, the wood-dwelling ones that still harbor the ancestral gut protists, the ones whose behavior most closely resembles what the first Jurassic termite colony must have looked like, are in decline. And every lineage that goes extinct takes with it a chapter of the story that no fossil will ever replace. There is something almost vertiginous about the full picture of what the DNA has Revealed. An insect we called a pest turns out to be a cockroach. That cockroach turns out to be the architect of some of the most sophisticated collective engineering on land. Its colony turns out to have begun 170 million years ago in a Jurassic forest. Millions of years before we had any fossil evidence of it. Built around a shared gut microbiome that co-evolved with the colony across every generation since. Its soldier caste turns out to be a developmental arrest, evolution reaching into the metamorphic toolkit of an insect and freezing it partway through to produce a weapon. Its queen turns out to be a chemical broadcaster whose molecular signals define the identity of every other individual in a colony of millions. And the workers, those small, blind, irreplaceable creatures, turn out to be carrying inside them the full genetic potential for everything the queen is doing. Suppressed, waiting, ready. The termite was not a simple thing that stayed simple. It was a complex thing that most of us never looked at long enough to see clearly. In the Namib Desert, the mounds are still standing. They were there before the Portuguese arrived on that coast, before the Mali Empire rose and fell, before the Roman world knew Africa existed. Some of those mounds are more than 1,000 years old, built by creatures whose individual lives last a few months, sustained across the centuries by the unbroken relay of a colony that has never stopped rebuilding itself from the inside. The queen inside is not the queen who started it. She died decades ago. But the pheromone system she ran, the molecular broadcast, the chemical definition of what each termite is permitted to be, that system was inherited and restarted and passed on through the colony's own internal logic, generation after generation. We did not build anything that lasts 1,000 years with blind workers the size of rice grains. We never learned how, and we are only now reading the genomes of these ancient social cockroaches, beginning to understand what they figured out 170 million years before we existed. The DNA is only beginning to tell us what it knows. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.